So thanks everyone for joining us at the symposium this morning. I wanted to start with gratitude and acknowledgements of the people who made this possible. First, I want to say that I stand today on Chochenyo Ohlone lands, and members of the Moeka Ohlone still live in the East Bay, and they keep their language and culture alive. And I'm grateful for their continued presence and efforts. And I encourage you all to learn more about the history of the lands that you are on at native-lands.ca. So pulling together a symposium is no small amount of work. And Tracy Misiewicz was essential in pulling together the speakers and the program. And Anne-Marie Benz, Maya Argaman, and Elizabeth Kuby worked hard on today's logistics to ensure everything runs smoothly. Our organizing committee of Daniel Blusenkamp, Brent Mishler, Bruce Baldwin, Heath Bartosh, Holly Forbes, Joyce Mashinsky, Christina Schierenbeck, Naomi Fraga, and Stu Weiss contributed their vision and experience to make today what it is. And I also want to thank Victor Schaff, owner of SNS Seeds, Hedgerow Farms, and Pacific Coast Seed, for his generous gift to CNPS to help us explore questions around seed provenance and to promote best practices for his industry. I also want to acknowledge the recent passing of John Anderson, another founder of California's native seed industry. So 2020 is the last year of the UN's Decade on Biodiversity, and 2021 begins the decade of restoration. Excuse me. The world is changing at a rapid pace, and California is no different. Climate change, sea level rise, catastrophic wildfire, and development contribute to threaten the foundations of biodiversity across the state. The governor's recent executive order exhorts us to protect and restore biodiversity. The actions we take, such as translocation of species and industrial scale use of native plants in gardening, landscaping, and restoration, can ameliorate or exacerbate prior harms. As a science-based nonprofit, California Native Plant Society contributes to solutions by synthesizing the best available science, by highlighting unknowns and potential issues, and formulating policies and recommendations to conserve California's diverse plant heritage. Given the urgent threats our native plants face, the growing interest in undertaking ambitious responses, and the level of risk involved in moving native species around the state without being able to unmix genes, it is time to bring together the best available science and start from the precautionary principle. First, do no harm. We welcome you to participate actively in today's symposium, to share your ideas and questions regarding genetic considerations in native plant gardening and restoration. The morning will explore California's floral diversity and some of the implications of moving species around. In the afternoon, we will delve into some of the more practical considerations in a breakout session after lunch, and a closing panel with plenty of time for questions and answers. And I'm pleased now to turn over the virtual microphone to Dr. Brent Mishler. Thank you.
Thank you very much. And welcome to everybody. We're so happy everybody joined in on such an important topic. And I'm pleased to be allowed to just give a general background of the matters and some of the reasons why we care about biodiversity and really what biodiversity is. So in these few minutes, what I'm going to do is, next slide, please. Next slide. What I'm going to do is point out that among non-scientists particularly, but even among scientists, there, go to the slide before. There we go. Thank you. That we take an overly typological view of things, and we consider that biodiversity is basically just species, and that's little packets that it's in, and if something's a species, everything inside that packet's interchangeable. And this is a very old view going back through the ancient Greeks and Christian theology, and is really not what we now think biodiversity is. What we now know biodiversity is this blooming and buzzing confusion of the tree of life, with lineages departing from each other and coming back together again from time to time. There are cutoffs that we arbitrarily impose just to try to get some order on things by calling something species and some things genera, some things subspecies. But there's nothing magical about any of those levels. Those are human constructs. The real thing is lineages. And we're going to hear a lot about those today. And I'm not giving specific examples. Bruce Baldwin, following this talk, is going to give some examples, and there are many other good examples during the day today. But I just want to get across a couple of take-home messages that species are really not that important. They're not comparable among lineages. As Darwin pointed out, they're just an arbitrary cutoff for convenience somewhere along a branch of life. That's different enough, so we'll call it a species. So when we're doing conservation, restoration, anything with biodiversity, we need to look at the whole tree. So now we can do the next slide, please. So this view was really the whole point of Darwin's Origin of Species book. Even though it mentioned species in the title, the whole book was about how species is just sort of an arbitrary level that lineages reach at some point, and that what really matters is the processes that are affecting them splitting, sometimes coming back together again, as we'll also hear about later in today's talks. So there are important lineages below the level that we traditionally apply the species label to, and definitely above that level. There is no sense in which organisms that bear the same taxonomic rank should be considered interchangeable in their genetics or their traits or their ecological preferences. So this has obvious implications for conservation biology and restoration. We can't just assume that because a plant has the same name as a plant that used to grow somewhere, that it has the same properties or would be a good replacement for what used to grow there. Next slide, please. The alternative, and I don't have time to go into this in any kind of detail, but I'm just going to present it as what you would do if you didn't want to worry so much about the taxonomic ranks and you wanted to actually measure biodiversity directly. You'd use a different measure that's called Phylogenetic Diversity, which just refers to how long is the branch going to the entity that you're looking at. The length could be measured in trait change, so it could be amount of genetic diversity that has arisen in one lineage since it split from its closest relative. It could also be time. You could measure these branch lengths in time. In this example, the branch lengths are measured in amount of genetic change. You see they're all different lengths and they're not coming to the present at the same time. And this is trying to make the point that if you had 14 terminal entities, in both the case on the left and the case on the right, you have 14 of these. These could be species or subspecies or lineages. You have much more diversity present in the right-hand situation than you do in the left-hand situation, where the 14 are each other's really close relatives. If they're scattered across a tree, basically phylogenetic diversity, what it does is it adds up those green branch lengths and comes up with a number, which is the path through the lineages that connect the entities that are at some particular point. Then there is a related measure called phylogenetic endemism that we use a lot, which is the inverse of the range size of a lineage. So if it occurs over so many square miles, 1 divided by that number of square miles is the endemism that it has. Next slide. Bruce and I and others have published a number of papers on this. I don't have time to go into it in any detail, but they are available as open source publications linked to from the University and Jepson Herbaria website. If you have interest in this, you can go click on that and go read the actual papers. But the idea is to switch from using taxa to using lineages directly and measuring biodiversity in the way I just mentioned by putting the tree of life right down and using it as a measuring stick, basically. And this is called Spatial Phylogenetics, which is a new big data sort of a thing that's only possible now because we're beginning to understand distributions of lineages better, and we also have better phylogenetic trees and better data and better tools to build them and study them. Next slide. These are from that paper that's listed at the bottom, and this paper is open source, and then, like I say, you can click to it on the Herbarium web page, the news page. I don't have time to sort of explain this, but you can then do randomization tests, which allow you to look at statistical significance of the patterns you see on the landscape. This one is a very high level comparison. This is looking at the whole state of California and very large scale lineages, but the same process could be used at any scale, could be used within one valley. These could be gene lineages in a very fine scale. So the scale doesn't matter. What matters is trying to understand how the lineages are occupying the landscape. And do they tend to co-occur with close relatives? Do they tend to co-occur with distant relatives? Are there concentrations of really long evolutionary branches in some places and not in other places? And that's really the goal of all this. Next slide. And these are from, the one on the left is from that same paper. This is a measure on the left-hand side there of the centers of really high endemism, and whether the endemism consists of really long branches or consists of really short branches. So it's paleoendemism and neoendemism respectively. And then the paper on the right is also linked to from the herbarium's page, is a shifting gears a bit. This is an assessment of starting where we are right now, what would be the top conservation priorities to move forward with. So this is using the principle of complementarity of what we already have conserved of the tree of California plants, and which parts of the landscape should we move forward on. And we measure this based on different interpretations of the branch lengths. As I mentioned, we can have branch lengths as in the blue up there that are scaled to time. As in the yellow, we can scale them to the number of genetic changes, or we cannot scale them at all, as in the purple on the bottom, which just basically shows how many divergence events are represented in a particular place, which can be useful as a measure. Next slide. And final slide. So the message here, take-home message was, let's look at the whole tree of life. Let's not worry so much about what's called a species or a subspecies, and instead worry more importantly about what it's related to and how distantly related to its closest relative it is. Particularly in plants, there's, as we'll hear a lot about today, there's a lot of fine scale variation within named species. That occurs in animals too, but particularly in plants. And this in plants, because of their modes of reproduction, is often highly correlated with geography, which is why many of us use geography as a proxy if we don't have phylogenetic studies fully available. And then the geography itself is part of the process, part of the evolutionary and ecological process that affects how biodiversity is generated and maintained. Therefore, as you'll hear a lot about today, plants are often locally adapted to the region or the climate or even the microclimate where they are. And so for all these reasons, this is the motivation for the symposium today. It's important to look carefully at distinct geographic populations and lineages, even if they're not named taxonomically, to avoid introducing plants that may not be well adapted there, to take advantage of the evolutionary experience that lineages have had in a particular environment, and so forth. And there I'll finish, Bruce. I made it in 10 minutes. So I now introduce Bruce Baldwin, the Jepson curator, who is going to continue.
Yeah, thanks, Brent. I'm going to pick up on one of the themes Brent mentioned, and that is the importance of recognizing and conserving fine scale diversity in the California flora. And next slide. So the amount of undiscovered plant diversity in California is unknown, of course, but based on evidence that's been compiled, it appears that the rate of description of good species, subspecies, and varieties hasn't really begun to abate. The graph in the right half of this diagram shows that over the past 100 years or more, we've had a pretty steady rate of description of new minimum rank taxa. When this was published in 2000, it was projected there'd start to be a tapering off of that description, but in fact, that hasn't come to pass, which is both exciting and humbling about the job ahead of us. And some of these discoveries have been plants, well, prior to the publication of this graph of the Shasta snow wreath, for example, which was discovered in 1992 near a major highway, and which was obviously new upon its first collection. Its closest relative still living is in Alabama, so a real paleoendemic. But oftentimes the plants that are described as new to science were already previously collected. If we can go to the next slide, please. And some of this would be in the category of what we can call cryptic diversity, which I define as distinct evolutionary lineages warranting taxonomic recognition that are difficult or impossible to distinguish morphologically. And you might say, well, that actually applies to quite a bit of what's already described and recognized in the California flora if you've had trouble keying plants out in diverse genera, for example. And that's true because we have a very young flora, very recent evolutionary divergences. And so it's not surprising that we'd have a lot of this diversity within California, including diversity that's not yet recognized. And this diversity represents ancient irreplaceable lineages that may not be ecologically interchangeable and that are critical to consider in conservation planning. For example, you might have distinct climatic adaptations, soil adaptations, et cetera. And I just want to go through a couple of examples. Next slide. These are both from the most diverse family of California plants in terms of numbers of recognized species, the sunflower family. And I could draw from many different groups of plants, just about any major lineage, major clade of California plants that people have investigated with molecular phylogenetic approaches has yielded evidence of new diversity, even if they were studied intensively before. And Lasthenia californica is such an example, the common goldfields, which in its old circumscriptions, arguably the most abundant native composite in the state, carpeting grasslands, foothill woodlands in the spring times through much of the cismontane California. And it's long been known that there was quite a bit of biochemical and morphological variation within this common species, which when originally described, recognized was an annual species exclusively. Next slide, please. But it has been studied now from the standpoint of molecular phylogenetics, from nuclear and chloroplast genomes. And from both genomes, we have similar evolutionary relationships that have been reconstructed, indicating that what we've called common goldfields in the past is actually two semi-cryptic species that are not each other's closest relatives, as the arrows indicate in this phylogeny. This is work by Raymond Chan. And you can see from the photos of these florets that you can tell these plants apart by their pappus elements, but only if pappus is present, and it's often absent in both, in which case they are truly cryptic, as far as we know, morphologically indistinguishable. Can I go to the next slide, please? Both of these now are, these lineages are recognized in the Jepson Manual 2nd edition as good taxa. You can see their names there, Lasthenia californica, subspecies californica, and Lasthenia gracilis. And even though their distributions are offset somewhat, they overlap extensively in Northern California. But interestingly, where they occur together, they're ecologically distinct. Can you go to the next slide, please? And this is a common theme we see in these cryptic taxa. So if we go to the next slide, thank you. Work by Nishi Rajakaruna in collaboration with my lab back quite a few years ago now showed that where these grow together, they occur in different soils. For example, serpentine versus non-serpentine or saline versus non-saline soils. Oops, back again. And these two different soil endemic groups were thought to be different races, ecological races. Can we go back one slide, please? Ecological races of the same species, but it turns out that each of these two taxa recognized now includes both of these races. That is, these different ecological types have evolved independently in both of these groups. Whoops. And Jen Yost, who's shown in the photo, we can go forward again, also at Cal Poly nowadays in San Luis Obispo, showed that at Jasper Ridge and other locations where these two taxa grow together, they show strong evidence of differential adaptation, for example, when you do reciprocal transplant experiments. So this is a great example of cryptic diversity, both on a large geographic and fine ecological scale, that's really important to keep in mind in conservation planning. Okay, so I'd like to go to the next slide. My last example is from the tarweed subtribe Medione. That's been the main focus of my research. And in the genus Leia, these spring ephemerals, Leia gilardioides, the woodland Leia, this is a group that was studied extensively by the Klozenkek and Heisey experimental biosystematic ecological team back in the 30s and 40s, and they noted very spectacular differences between populations of this species as it was recognized, and still is, and it's incidentally, pending further taxonomic efforts in my lab. But you can see that the flower color variation can be striking. Go to the next slide. You can see more significant variation in leaf lobing. So these are basal leaves, silhouettes, individual leaves from different plants from 3 coastal populations on the left and 3 interior populations on the right. And you can see that the lobing is greater in the coastal populations, or sorry, in the interior populations than in the coastal populations. Klozenkek and Heisey attributed this to local adaptation to maritime versus continental climates, with deeper lobing being associated with more xeric climate. But in fact, we found that this represents 3, as the colors indicate, different major evolutionary lineages or clades that are quite ancient. If we go to the next slide, you can see here how they replace each other in the lower right on a coastal to interior transect. They turn over this very steep climatic gradient as we go from the coast to the interior. These colors reflecting the colors on the clades on the tree on the left. And you can see that that coastalmost clade is actually more closely related to 2 other species flagged by those thick arrows, Leia harasioides and carnosa, than it is to the other members of Leia gilardioides and the way it's been treated. So we're in the process of breaking this up taxonomically. There's also a Humboldt County clade there in purple that wasn't previously studied, but much more diversity here than previously indicated. And I also call this cryptic or semi-cryptic diversity, because even though the basal leaf morphology is very nicely indicative of which clade you're dealing with, those leaves are withered generally at the time the plants are in flower, and the plants are generally collected and recognizable as Leias when they're in flower. So effectively, they are fairly cryptic, but ecologically very distinctive. Okay, so we go to the next slide, please. So among other tar plant genera that we've studied, we find example after example of cryptic diversity within these. Deonander bacchiglupii is a species there at the top center that we've described from alkali scalds in Livermore Valley that was previously treated as part of Deonandra increscens, very distantly related within Deonandra. And within these other 5 species here, we have cryptic diversity, sometimes more than a single entity awaiting description. And we're getting around to this hopefully shortly, but 3 of these are already considered CNPS 1B taxa. So quite critical to bear in mind that even rare plants can have cryptic diversity within them that's important to consider. Next slide, please. And finally, another layer on all of this is that you can have cryptic obligately associated taxa, in this case, animals, heliothione moths, that in the case of tarweeds were thought to represent a couple of species in 2 genera, Heliothodes and Microhelia, but work by Terry Sears and others now following up, at least 19 known tarweed specialist moths that can be told apart by fine details of their wing patterning. It's beautiful moth wing patterns but previously unrecognized. And these are endemic to particular clades of tar weeds, some of which are cryptic themselves. So we have cryptic diversity layered on cryptic diversity, and it's quite humbling and quite exciting, but it shows what we're facing in terms of the magnitude of biodiversity discovery in the California flora. So final slide, please. So in conclusion, plant taxonomy in California is still very much a frontier. And for that reason, species or subspecies and varieties should not be regarded as ecologically or evolutionarily uniform, as Brent mentioned earlier. And loss of this diversity, even if it's undescribed, of course, is extinction. We're not just talking about loss of genetic variation, we're talking about extinction of ancient lineages, just as much extinction as extinction of any named taxon. And this can take with it associated organisms like the heliothione moths, for example, all of which are irreplaceable groups. And so the lack of detailed data on the lineages that exist within most Californian plant species warrants their conservation across the ecological and geographic ranges of those species, and cautious mitigation, which is critical when considering irreversible actions, as Andrea mentioned, the importance of the precautionary principle. And we'll be hearing more about cryptic diversity here shortly from Lee Johnson, but I'll leave it at there. Thanks very much.
Thanks, Bruce. So it sounds like Justin has some more information about cryptic diversity in the goldfields. Justin, did you want to add that comment as well?
It says I can't start my video, but hey, Bruce, that was awesome. And I wanted to mention that Jen Yost and her students have been identifying cryptic UV patterning in the ray petals now, which is only seen using UV photography, which just points to new technology, maybe revealing additional lineages, just like your ITS sequences revealed additional lineages. And we just can't predict what future technology will hold.
Great point. And, you know, there are those two new taxa recognized, but there definitely is additional lineage diversity within those groups as well that hasn't been named, but it's worth keeping in mind. Yeah, I was fortunate to be the outside member on Jen's dissertation, and she had that the data on that cryptic patterning from UV back then. It's really exciting stuff. Thanks, Justin.
Great. Thanks, Justin. And it sounds like Stu may have had a question as well.
Okay, yeah, I'm unmuted. Yeah, my question actually was in the, someone asked it already in the Q&A for later discussion about reconciling the Endangered Species Act with this new view of biodiversity.
Okay, yeah, we'll talk more about that in the rare plant breakout session this afternoon. So we are at a break time now, and I wanted to thank our speakers for the opening plenary and thank all of you for coming.